Is everybody comfortable? Got their scribble pens out. So hopefully my migraine is gone enough that I can be coherent today. Took some drugs. Hopefully we're feeling good. Because so I had 4 lab tours of high school students in my lab this afternoon for 20 minutes at a time. It's not so bad, but definitely you got to be a little more excitable than for you guys because they're like not all that. I'm like, so what are some macromolecules you can think of? Glucose? Like, let's go a little bigger. So anyway, so we tried to talk about proteins and enzymes. And anyway, and then this news camera came in for like News Channel 14. I'm like, oh, seriously? So I think perhaps at 5 o'clock today, there might be some footage from my lab. I think she was filming the students, not me, so that's good. Okay, so we're going to dig into metabolic regulation today, and so hopefully this will be a nice review of the enzymes we've learned about, and especially the enzymes that are metabolically irreversible, so the control points in glycolysis and gluconeogenesis. So you'll see at the end of this we get into sugar breakdown and synthesis. We may not get there today, but that's also regulated. So it's all part of sugar metabolism. Okay. So when you think about what are the products of glycolysis, so of course one of them is ATP, right? The other is reduced cofactors, in particular NADH. But if you think about, if you had to sense what the metabolic state of the cell was, you could think about how much ATP is around. So if you have a lot of ATP, that means you're in a good place metabolically, energetically. If you have not so much ATP, then you might want to do some glycolysis. So we're also going to see that AMP is actually a better sense of where you are metabolically than ATP. So these are just some kind of overviews that you may want to go back to later, but we're not going to talk about the details now. So we're just kind of at it. So the point is, we could think about what's our ATP level. However, ATP levels don't actually change that much. Okay, so if you think about, so what are some activities that might deplete ATP? Okay, gluconeogenesis, running. Okay, doing some kind of exercise. So muscle movement is going to use ATP. Certainly plenty of other things are too. But if we think about, we have to get up and walk somewhere. Okay, so you're going to use ATP. So if you're not eating, so if you kind of could shut down fuel consumption and fuel usage, and you just had to work on the ATP that you had already in your system, that would go down a little bit. But it's still not going to go down very much. Because you still have plenty, fortunately, we have plenty of ATP. We're going to be making DNA or anything else. And so our ATP stores are not going to be depleted dramatically. So it's going to go down a little bit, but it's really only going to change by 10%. Okay, because there's going to be lots of enzymes that will work to make ATP, especially, so if you use a molecule of ATP to do something like muscle contraction, the product of that is going to be ADP. ADP doesn't really do us much good, so the enzyme adenylate kinase will recycle 2 molecules of ADP to get back to a molecule of ATP and make an AMP. Okay, so it's just a way of maintaining ATP levels because we're going to need them. ADP doesn't do us any good, so we're going to make some ATP. So really, it's AMP that's going to change the most dramatically. Okay, so ATP doesn't change very much. DP doesn't change, AMP goes way up. So really we shouldn't be monitoring AMP as an indicator of we need to make some more ATP. Does that make sense? So if you're going to monitor something, it's better to monitor AMP. Okay, so how does AMP get monitored? So there's a protein, an enzyme called AMP activated protein kinase that's going to globally do lots of things in lots of tissues. Okay, so this enzyme, so we know, so if it's called kinase, what is it going to do? Phosphorylate. It's called protein kinase, so it's going to phosphorylate proteins. And as we'll see, phosphorylation of proteins is a metabolic regulation mechanism. Okay, so we're going to phosphorylate proteins that are going to help us get more ATP. Okay, so the downstream effect of having AMP is making more ATP, because AMP is the signal that we need more ATP. Okay, so there's a whole bunch of anabolic pathways that get turned on, catabolic pathways that get turned off. So you can just look at a couple of these as examples. So for example, there's a signal in your brain that regulates food intake and fuel metabolism. Okay, so in fact, rising AMP levels, activating this AMP-activated protein kinase will phosphorylate proteins, which will lead to a signal in your brain that says, I'm hungry. Okay, so if you're low on ATP, high on AMP, you might want to intake some more fuel, so you might want to get something you can feed into your metabolic pathways to make more ATP. Okay, so food craving might be a signal that you're low on ATP or high on AMP. Okay, so that's one example of upregulating. So again, these little green triangle means upregulate. The red X's mean downregulate. Okay, so would it make sense to downregulate fatty acid biosynthesis? Yes. Okay. And so let's just think of lipidolysis. So breaking down lipids. So if you have high AMP, why would it make sense to inhibit lipid metabolism? Katie. Well, if you have high AMP, that means you need more ATP.
Oh, weird. I thought you were saying, did you say lipids?
So lipidolysis. So breaking down lipids to use for lipid metabolism.
You would use your lipids, correct?
Right. So you would use your lipids for energy. Okay. And so yeah. So you're gonna upregulate lipid metabolism and food intake and downregulate storage. Okay. So you're gonna stop storing fuel and keep using more fuel. Okay, so this is kind of a more advanced summary slide of one that we talked about when we talked about enzyme regulation of activity. So there's lots of ways that we can regulate an enzyme activity. So one is we can just make more of it. We can downregulate enzyme activity if we chew up the message or if we chew up the protein. We've talked about allostery. We've talked about phosphorylation. So we're going to see lots more of those. So again, just an overview. Okay, and again, we've talked about the steps that are separate in glycolysis and gluconeogenesis as being the ones that we have to regulate. So again, the 3 reactions of glycolysis that we have to bypass in gluconeogenesis. So those are the ones we're going to focus on. So another way, what are the 2 classifications of metabolic enzymes that we've talked about? So enzymes can be, okay, that's one way we've categorized them. What about in terms of, that's what I was thinking of. Okay, so near equilibrium or irreversible, metabolically irreversible. Okay, so I'm going to give you 2 other ways of describing them, which mean the same thing but come at it from a different way. So if a metabolic enzyme is near equilibrium, that means if we give it a little bit more substrate, it's going to go faster. Okay, so increase the flux through that pathway if you have more substrate. So you see how those are kind of the same thing? So if you have an enzyme that's metabolically irreversible, will adding more substrate make it work faster? Not really. Okay, so if it's metabolically irreversible, it's not near equilibrium. So increasing the substrate concentration really isn't going to do much. Okay, we'll see an example of that in a minute. But how would you make the overall reaction go faster? You could add more enzyme. Okay, so if each individual enzyme molecule isn't going to be improved by adding substrate, we could just make more protein, add more enzyme. Okay, so another way of describing those are substrate limited equals near equilibrium, or enzyme limited means it's already going as fast as it can given the conditions. So the only way we could make that overall step of the reaction go faster is if we put in more enzyme to the mix. Does that make sense? Okay, so rate limiting or enzyme limited, we could also call those metabolically irreversible. Okay, so if you look on the right, so the blue reactions, we would call those near equilibrium. Okay, so we could make them go faster simply by adding more substrate. The yellow enzymes, okay, so the yellow steps would be enzyme limited. Okay, so see how intermediate E is kind of piling up? So that reaction is not going to go faster simply because you have more substrate. It's kind of going as fast as it can. If you now made more enzyme, now you would increase the flux through that step of the path. Does that kind of make sense? So we'll come back to that. So just kind of let that percolate for a little while. We're saying kind of the same thing. Okay, and so on the bottom left, so if you look at the x-axis, so look at the y-axis. So that's glycolytic flux. So how fast we're moving through glycolysis. So higher up on the y-axis means we're kind of pushing through and getting more end product, more pyruvate. And on the x-axis, that's how much enzyme there is. So for each of those three enzymes, so for example, phosphohexose isomerase, at a given substrate concentration, we could add more enzyme and it's really not going to do much of anything. Okay, so that's not an enzyme-limited step. However, phosphofructokinase 1 is to some extent enzyme-limited. Hexokinase 4, definitely enzyme-limited. Okay, so if we add more enzyme, we'll get more end product of glycolysis. Does that kind of make sense? Will it help a little bit? Okay, again, we're going to come back to these. We're just kind of setting the stage for our discussion on regulation. Okay, so, yes?
Oh, so the lower the delta G, does that mean it's more affected by?
Enzyme limiting. Yep.
Okay.
Yeah, so those very metabolically irreversible reactions, so they're not controlled by how much substrate you have. They're controlled more by how much enzyme you have. Yep. Okay, so I'm going to introduce the idea of isozymes. Okay, so isozymes are enzymes that do the same chemistry, but they're different polypeptides. Okay, so they have distinct genes. So distinct sequence, but same chemistry. Okay, so for this example, hexokinase and glucokinase both catalyze the phosphorylation of the C6 of glucose to make glucose 6-phosphate. Okay, so we've talked about that enzyme as hexokinase, but there's also an isozyme that's called, so the one that we have talked about so far is called hexokinase 1. Its isozyme hexokinase 4 we also call glucokinase. So I'm going to stop saying hexokinase 4 and call them hexokinase and glucokinase. Okay, so hexokinase is the one you learn for glycolysis, but there's also another enzyme, glucokinase, that will phosphorylate glucose to make glucose 6-phosphate, but it's a different polypeptide. Okay, so glucokinase is in the liver. Hexokinase is in everywhere, but we're going to think about it as being in the muscle. So to simplify, we can say hexokinase does glycolysis in the liver. Hexokinase does glycolysis in the muscle. Glucokinase phosphorylates glucose in the liver. Muscle, liver. So hexokinase does glycolysis in the muscle. Glucokinase can do glycolysis in the liver, but it can also do other things. Does that make sense? But the same chemical reaction. Different polypeptides. So the advantage of having different genes, different polypeptides, is they can be regulated differently. Okay, so we can tweak where glycolysis happens based on regulation. Okay, and so how are they regulated? Okay, so in the liver, we can do glycolysis, but we can also do other things. Okay, so we can also make glycogen. So remember, glycogen is in the liver, so it's used in the liver, and it's also made in the liver. Okay, so extra sugar that we don't need for glycolysis can be stored as glycogen. That happens in the liver. So phosphorylation of glucose to begin the process of glycogen storage happens in the liver using a glucokinase. Okay, so would it make sense for them to be regulated the same way or to be controlled separately? Separately. Okay, so under what circumstances would you want to be making glycogen? So you have an excess of glucose, you have all you need for fuel, and you have an extra. So it might be that you have extra glucose that you want to store. It might even be that you have plenty of fat to burn as fuel, and you're just going to go ahead and store the extra sugar as glycogen. Okay, so they're going to be regulated differently. So hexokinase in the muscle, so that's this one here. So if you had to guess at the Km for hexokinase, what would it be? Very small. Okay, so we can't even hardly tell what that number is, but it's going to be, less than 1 millimolar. Maybe we'll say 0.1 millimolar. We're guessing. It's very low. What would you guess for the Km of glucokinase? So more like 5 or 6 millimolar, right? So the substrate concentration that gives you half the Vmax. So it's much higher for glucokinase. So why does that make sense? Based on what you just said. Jessica.
Oh, I was just thinking maybe because the hexokinase is specific for glycolysis, but the glucokinase can
Okay, so we only want to store sugar as glycogen when we have a whole lot extra of it. Okay, so we're not going to steal the glucose from the muscles if we need to do glycolysis to get ATP. So if you had a very low Km for glucokinase 2, there'd be these competing uses of glucose. Using it for glycolysis and using it for storage would fight each other. Okay, so having a higher Km for glucokinase means we're not going to store glucose as glycogen until there's so much of it that hexokinase is doing all it can and there's just extra left over. Does that make sense? Okay, so they're different polypeptides, so they can have different Kms. Okay, we'll also see that hexokinase is inhibited by its product, whereas glucokinase isn't. And part of that is because we don't necessarily want to inhibit glycogen formation while we're doing glycolysis as well. You can kind of ignore that for now. But the most important thing is they have different Kms, which means they're going to use the glucose under different circumstances. So glycolysis is always going on. We only do glycogen synthesis if there's a surplus of glucose. Everybody okay on that idea? More or less? Okay. Again, any of these we can come back to. We're going to kind of build as we go different layers of regulation. Okay, so another way that glucokinase is regulated, so again, this is glucokinase. And again, glucokinase, or hexokinase 4, is mostly in the liver. Okay, so here's a wacky thing. So where are proteins usually found? This is what I ask the high school students. Where would you find proteins? They're like, in the mitochondria? I'm like, yeah, but... So where do you find proteins in general? Cytoplasm. Okay. Where does chemistry happen? In the cytoplasm. So if you have proteins in the nucleus, sure, there's some proteins that do their work in the nucleus, but not a lot of chemistry happens in the nucleus as far as just enzymology. So if there's a mechanism for hiding glucokinase in the nucleus, that's going to inhibit its function. All the substrates are in the cytoplasm. Now the enzyme is not where the substrates are, so chemistry is not going to happen. Okay, so the other way that glucokinase is regulated, besides just having a high Km, is it's sequestered. Okay, so this is regulation by sequestration. Okay, and so how does that happen? So if we have sufficient fructose 6-phosphate, that means we're doing glycolysis. So it turns out that glucokinase is going to go into the liver. So in the liver, if glycolysis is happening already, we're just going to, get glucokinase out of the way. We're not going to need it right now. Okay, and it's only going to come out of the liver. Out of the nucleus if we have a very high glucose concentration. So remember, it has a high Km for glucose. So not only is it going to work more, get closer to its Vmax if glucose is high, but also it can sense, so glucose presence in the liver cell will basically pull it out of the nucleus. Okay, so it's there in the cytoplasm where it can do its work, and it's going to, at high glucose concentrations, do what it does, which is phosphorylate glucose to make glycogen or to do glycolysis. Okay, so it's hiding in the nucleus until glucose levels rise enough that it's needed. Make sense? Okay. So that's the second way that it's regulated.
If there's a lot of fructose, does it come out or come in?
So if there's, you mean fructose or fructose 6-phosphate? So if there's fructose 6-phosphate, it'll go back into the nucleus until it's needed, until glucose levels rise again. Okay, so it's kind of like feedback inhibition, but it's not inhibiting its activity, it's just inhibiting its presence. Okay? Okay, so that's regulation of glucokinase versus hexokinase. Okay, so hexokinase, remember, is the first metabolically irreversible step of glycolysis, and so that's how that enzyme is regulated. Two different ways, basically, the two different isozymes function differently for different purposes. Okay, so the next enzyme of glycolysis that's metabolically irreversible that we need to control, we need to regulate, is phosphofructokinase 1. Okay, so it's the committed step for glycolysis. So why is hexokinase not the committed step of glycolysis? Is there anything that glucose 6-phosphate does besides enter glycolysis? Yay, pentose phosphate pathway, right? Or glycogen synthesis. Okay, so we came to a fork in the road with phosphofructokinase 1, and the question is, are we going to keep going in glycolysis? And if we are, then we're all in, and if we're not, then we're going to go back to gluconeogenesis. Okay, so this enzyme being the committed step, it means, once we've made fructose 1,6-bisphosphate, we're going to keep going. Okay. So it is regulated by allostery. Okay, and so what are the allosteric modulators of phosphofructokinase 1? So certainly AMP and ADP will activate. So if we have high AMP, that's an indicator that we need to get more ATP, so we better do glycolysis. Okay, so in what way is ATP a negative modulator? So it's kind of feedback inhibition, right? If one of the main products of glycolysis is ATP, if we have enough ATP, why do we keep going in glycolysis? Let's shut it down or downregulate it. So we're going to see this molecule in a minute. Ignore it for now. So we haven't talked about citric acid, but from what you know about overall sugar metabolism, how is citric acid or citrate a feedback inhibitor? Right, so it means we went through glycolysis, we entered the citric acid cycle, so if we have sufficient citrate, maybe it came from glycolysis or maybe it came from fatty acid breakdown, but in any case, we're good metabolically, we don't need to keep going through glycolysis. So citrate is also a feedback inhibitor. Okay? So what do you see about the shape of that curve? Sigmoidal. So what do you know about phosphofructokinase 1? Okay, there's multiple subunits and there's cooperativity. So I showed you the picture over here. So hopefully you can see that's a dimeric enzyme. It looks tetrameric, but it's really just dimeric. Okay, so it's a dimeric enzyme. There's cooperativity between the subunits, which makes so it's not Michaelis-Menten hyperbolic curve. It's still an activity curve, but it's not hyperbolic, it's sigmoidal. Okay, so now you know that about PFK1. And so in the presence of ATP, everything is suppressed. So for the same substrate concentration, we have lower enzyme activity in the presence of ATP because it's feedback inhibiting, basically. And if we have low ATP, then we have more activity of the enzyme. Okay, does that make sense, the graph on the left? So you've seen that before for things like CTP inhibiting ATCase, right? Sigmoidal curve, multiple subunits. CTP is a feedback inhibitor, so you get kind of repression of that curve. So what do you think the graph on the right means? So this is activity of PFK1 with ATP concentration now. So that means, so just look at the solid line for a minute. So if we have high ATP, enzyme activity decreases. So that's saying the same thing as on the left. But to show you that AMP is actually more important than ATP, even if we're at high ATP, if we now spike in AMP, that signal is going to win. Okay, so the high MP is more of a measure of your metabolic state than how much ATP you have. Okay, so the AMP will dominate and kind of kick out the ATP. So I think on this structure, so I don't know if you can see those very well, I think that guy is the ATP. Well, actually, that's not true. That's not true. So here is, so this is the active site. So remember, it's a kinase, so ATP is a substrate as well, right? So ATP and fructose 6-phosphate. So those are the substrates to make phosphofructokinase 1. Sorry, fructose bisphosphate 1, right? So that's the active site with the substrates bound. And then the allosteric site is right there. Okay, so this is, the blue is ATP. And there's another ATP sitting right there that's not in the active site. It's not a substrate. It's an allosteric regulator. So how does that make sense if ATP is both a substrate and an allosteric regulator? Okay. Kind of, yeah. So you need some ATP to make the reaction work. So that's the substrate ATP that's going to bind in the active site. Now, if you have more ATP, now it's not only going to bind in the active site as a substrate, it can also bind in this extra site. And that will actually downregulate the enzyme activity. So you wouldn't want to not bind any ATP at all or have no ATP because the enzyme wouldn't work. So what would you guess about the affinities, the KD, for those two binding sites? So ATP binds at two separate places, active site and allosteric site. Which one's going to bind more tightly? Active site. Okay, so at low ATP concentrations, it's only going to go to the active site because that's where it binds more tightly. Now, as ATP levels rise, it's going to bind in the active site, but now it's going to also bind in the allosteric site and turn down activity. So you have to have a high enough concentration that it can bind in the allosteric site as well. Does that make sense? So if ATP is bound in the allosteric site, we're going to downregulate activity. If AMP is bound in the allosteric site, we're going to upregulate activity. So same binding site, but different effects on the enzyme activity. So you could, I'm not sure that people use R and T state to describe this, but you could, file it in a way in your brain that way. So ATP is going to shift towards the T state, AMP is going to shift towards the R state. Yep. Yeah, so I think there's only one allosteric site and it binds either ATP or AMP. Brooke, did you have a question?
I was just going to ask, so KD, so the KD for the active site is smaller?
Yes. Yeah, so you have a lower KD, so it binds at lower ATP concentrations. You need more ATP to bind in the allosteric site. Yes.
So would AMP have a lower KD than ATP?
Yes.
And does citrate bind there too?
I'm not sure, actually. Probably somewhere else. I mean, it's got to be an allosteric site, but I'm not sure if it's overlapping with ATP and AMP or not. Yes?
Sorry. Can you say what you said again about the T and the R?
Okay. So, again, I'm not sure that people talk about T and R states with PFK1, but the idea works.
Okay.
So if we say the T state is the less active state, then ATP binding will shift towards the T state. Okay, so if we have ATP, we don't need to keep doing this reaction. So we're going to shift to the T state, turn off activity, or turn down activity. If we have AMP, it's going to shift it to the R state, turn up activity. Because AMP means we've got to keep going in glycolysis, so we're going to keep going in this direction.
So the AMP
Well, so it's going to be sigmoidal no matter what. Oh, wrong way. So they're both sigmoidal. It's just that it's going to shift up in low ATP or high AMP. So it's going to shift up when you need ATP energy. It's going to shift down if you have sufficient ATP. But it's sigmoidal no matter what. Okay, ready to move on? Okay. So the complementary enzyme to phosphofructokinase 1 is fructose 1,6-bisphosphatase. So again, that's the gluconeogenesis enzyme. So that's bypass reaction 2 probably. Okay, so it's the first enzyme in glycolysis that we're going to deal with this way. No, so it's bypass reaction 2. So how might it be regulated? The opposite of phosphofructokinase 1. Okay, so those are opposing activities. So kinase and phosphatase are opposites of one another. So all the things that would activate phosphofructokinase 1 should probably deactivate fructose 1,6-bisphosphatase. So it turns out that the only one that is a direct comparison is AMP. Again, pointing out the significance of AMP. So AMP will activate phosphofructokinase 1 and inhibit fructose bisphosphatase 1. That's all we need to say there. However, okay, we're still on phosphofructokinase 1, on that reaction in glycolysis. So we're going to add in another layer of allosteric regulation. Don't let it bother you. Here, we're peeling back the onion here. Okay, so we're going to introduce a new molecule. So fructose 2,6-bisphosphate, this guy here. So does that sound like something we've just been talking about? So it sounds like fructose 1,6-bisphosphate, but it's fructose 2,6-bisphosphate. Okay, so the only difference is where that second phosphate is. Okay, and so the enzyme that makes that is, we'll get there. There it is. Actually, before we talk about the enzyme that makes it, we're going to ignore the enzyme that makes it for a minute, and we're going to talk about its effect. Okay, so let's just assume we're going to make it somehow. And fructose 2,6-bisphosphate, this molecule, is not an intermediate in glycolysis or gluconeogenesis. It's an allosteric regulator of both phosphofructokinase 1 and fructose 1,6-bisphosphatase. So there's two opposite enzymes. Okay, so it's not a metabolite, it's a modulator. And it turns out it modulates both phosphofructokinase 1, so here, so PFK1 activity. So in the absence of this molecule, activity is pretty low. Okay, so it's still sigmoidal, so you can still see that sigmoidal curve. So we're kind of looking at the very beginning of the curve. So at low substrate levels, if we don't have fructose 2,6-bisphosphate, we don't have much activity. If we do have fructose 2,6-bisphosphate, we do have a lot of activity. Okay, so this is a heterotropic positive modulator of phosphofructokinase 1. Dials up activity of PFK1. Yep. So fructose 2,6-bisphosphate is a heterotropic positive modulator of phosphofructokinase 1. So in the absence of it, not much is going on. In the presence of it, now we have robust activity. At the same time that it's a heterotropic positive modulator of phosphofructokinase 1, it's a negative heterotropic modulator of fructose bisphosphatase. Okay, so phosphofructokinase 1 is the glycolysis enzyme. Fructose 1,6-bisphosphatase is the gluconeogenesis enzyme. So they're exactly opposite of one another. Kinase phosphatase, the same molecule, turns up activity of one enzyme, down activity of the other enzyme. So it basically amplifies the difference in their activities. So both AMP and fructose 2,6-bisphosphate will amplify the difference in those enzyme activities. So why do we not want those two enzymes working at the same time at the same rate? It's counterproductive. It's wasting ATP. So phosphofructokinase 1 uses ATP to phosphorylate. Fructose 1,6-bisphosphate cleaves off that phosphate with nothing to show for it. It doesn't make ATP, it just cleaves off that phosphate. So if both of those enzymes are going at the same time, we're just chewing through ATP and not getting anything from it. We're just going around in circles. So if we have a molecule like fructose 2,6-bisphosphate that can upregulate one and downregulate the other at the same time, all the better for us. Okay, so we're amplifying the difference in those two pathways. Turn up one, turn down the other at the same time. Okay, so fructose 2,6-bisphosphate is a heterotropic negative modulator of fructose 1,6-bisphosphatase. Okay there? So both AMP and fructose 2,6-bisphosphate, heterotropic positive modulator of phosphofructokinase 1, heterotropic negative modulator of fructose 1,6-bisphosphatase. Okay, now we're going to get to how this molecule is made. Okay, so it's not an intermediate, it's just a modulator. Yes, Carter?
So does that just suggest that when glycolysis is happening, that deactivates gluconeogenesis?
Yes, absolutely. For many ways. So ATP will inhibit gluconeogenesis at several places. This molecule also is a major inhibitor of gluconeogenesis. Yeah, you don't want both pathways going at the same time. Liam, did you have a question?
So would you say they're like very similar? They do like the exact same thing on the same pathways? Like AMP and
Essentially, yeah. So AMP, ATP will act on multiple pathways in ways that make sense metabolically. You know, if you're going to turn down one, you're going to turn up the other. Yep. Okay, so how is this molecule fructose 2,6-bisphosphate made? Funny thing is it's made by a phosphofructokinase, but now it's phosphofructokinase 2. Oh, I know, it's awful. But isn't it convenient that phosphofructokinase 1 puts a phosphate on the C1? So where do you think phosphofructokinase 2 puts the phosphate? C2. Okay, so at least it makes sense. Okay, so we start with the same molecule, fructose 6-phosphate. So it has a choice. It's either going to be used by phosphofructokinase 1 that will keep going in glycolysis, or some little bit of it, not much, some small percentage can get shunted off to phosphofructokinase 2 and make fructose 2,6-bisphosphate, which will then amplify, turn up glycolysis, turn down gluconeogenesis. So basically you're wasting a little bit of your substrate, but in order to maximize your metabolic efficiency. Okay, so phosphofructokinase 2 basically steals a little bit of fructose 6-phosphate, but to make this regulator that will turn up glycolysis, turn down gluconeogenesis. Okay, so because there's a phosphofructokinase 2, there's a corresponding phosphatase, fructose bisphosphatase 2. Makes perfect sense. Kinase, phosphatase, working hand in hand. What doesn't make sense, or wouldn't be obvious from the beginning, is that phosphofructokinase 2 and fructose bisphosphatase 2 are the same protein. Crazy. Okay, so you have a kinase and a phosphatase, so there's actually two active sites, but they're on the same polypeptide. So one polypeptide can do two opposite functions. Put the phosphate on, take the phosphate off. I told you it was going to get messy today. So does anyone need a stretch break? I know we're like deep in. Do you need to get some oxygen? And no. I mean, not draw it. You would never have to draw it. So I might, I could give you the structure and ask, why is it useful for, or how does it bind to DNA? Why is it useful for seeing DNA? Yeah. Okay, let's come back. Okay, so fructose 2,6-bisphosphate is made by this new enzyme that's not a glycolysis enzyme. It's off to the side. So phosphofructokinase 2 uses a molecule of ATP to put a phosphate on the 2-carbon. Fructose bisphosphatase 2 works in opposition, we'll take it off. Okay, same polypeptide, two active sites, two opposite activities. Kind of weird. So you wouldn't want them both to be working at the same time, so we have to regulate this enzyme.
It won't add that extra phosphate to ADP or anything. It just releases it.
Right, it just releases it. Yeah, it just clips it off. We're not going to get anything from this. Yep, so same thing as phosphofructokinase 1, fructose bisphosphatase 1, which are two separate proteins. This one now is the same polypeptide, two active sites. So it's almost like you fuse those two proteins together into one polypeptide. So if you had both of those working at the same time, that would be a futile cycle, so a waste of energy. So how do you think they're regulated? No, so they're not regulated allosterically, they're regulated by covalent phosphorylation. So now our regulator molecule is regulated. So that's where we're digging deep into regulation. Okay, so the same polypeptide, if it's not phosphorylated, the kinase part is active. If it is phosphorylated, the phosphatase part is active. Okay, so with one event, we're going to turn on one activity and turn off the other, so you would never have them both working at the same time. So it's all or nothing. If it's phosphorylated, it's a phosphatase. If it's not phosphorylated, it's a kinase. One or the other, nothing in between. So if it's phosphorylated, the phosphatase is active and it plucks off that phosphate from the 2 carbon position. If it's not phosphorylated at that tyrosine or whatever it is, I think it's a tyrosine. If there's not a phosphate group on that residue, then the kinase part of it is active. The phosphatase part is not active. All or nothing. And so if the kinase part is active, what does it make? Fructose 2,6-bisphosphate, which upregulates what? By what enzyme does it upregulate? Phosphofructokinase 1, which directs towards glycolysis. It also inhibits at the same time fructose 1,6-bisphosphatase. Okay, so making of this molecule will upregulate glycolysis, downregulate gluconeogenesis. So if we phosphorylate the protein, we're going to upregulate gluconeogenesis, downregulate glycolysis. Okay, so phosphorylation equals inhibits glycolysis, stimulates gluconeogenesis. Because phosphorylation means we're plucking off the phosphate from the 2 position, we're degrading fructose 2,6-bisphosphate down to fructose 2-phosphate, fructose 6-phosphate. Okay, so if the kinase part is active, we're making more fructose 2,6-bisphosphate. We're upregulating glycolysis, downregulating gluconeogenesis. Okay, so peel back another layer. What's going to control whether this polypeptide is phosphorylated or not? Hormones controlling another kinase at another phosphatase. Crazy, I know. Yes, Carter.
So like for diabetics, when they have a glucagon shot, I guess that's what it is?
Insulin shot.
Is that glucagon? Is that what it is?
So,
It is. So that.
do diabetics ever have glucagon shots? Oh, so if their sugar is
Sugar's like super
too
low.
low.
Yeah.
Ah, okay. So let's think about for those of you who haven't thought about that before, you got one in your bag. Okay. Some people just have a shot of orange juice. Yeah.
Icing.
What's that? Icing? Oh, that's
Because you
good.
just absorb it in your mouth.
Yep. It'll make my mouth water thinking about it. Yeah. So insulin is the hormone, and what does it indicate? Not what does it do, but what does it indicate? High blood sugar. Okay, so the downstream effect, if you don't have diabetes, is bring the sugar into the cell, use it for glycolysis. Okay, so if we have high insulin, insulin will upregulate the phosphatase that takes off the phosphate from this bifunctional protein to turn on the kinase part to make fructose 2,6-bisphosphate to upregulate phosphofructokinase 1 to upregulate glycolysis, to use the sugar that's high in your bloodstream. And I think I made no mistakes in that sentence. Aren't you proud of me? It's hard to do. Yes, Carter.
So if you were to have a shot of the glucagon, would that just stop your body from processing sugar?
Essentially. Okay, so let's get to glucagon now. So we talked about insulin. So what does glucagon indicate? Not what does it do, but what does it indicate? Low blood sugar. So what are some of the things that you might want to turn on if you have low blood sugar? So if glucagon senses low blood sugar, what might the response to that be? Gluconeogenesis. Right. Okay, so why would you need to make glucose? What part of your body needs glucose? The brain. Okay, so if you have low blood sugar, you might be able to process fat for your muscles, but your brain needs the sugar. Okay, so if anyone wants to volunteer what happens when they have low blood sugar, what happens to your brain? Can't think. Okay. You start what?
I like to make funny. I try to make funny jokes.
Okay.
Don't make any sense.
So kind of like now. So from what I've heard from my friends with diabetes is the initial states of low blood sugar looks a lot like you're drunk. Like you're making a little bit less sense. You maybe seem a little sillier, or like I can't quite focus, maybe, but I can't really explain why. Like you're feeling okay, but something's not quite right. And then if it stays low long enough, so you get hungry, yes, because that's your body saying, "Whoop, got to adjust to this somehow." But if you stay low long enough, then what happens? Pass out. Right. That would be bad. Hopefully you've never gotten there. Good. Okay, so ultimately, your body kind of says, wait a minute, we're in crisis mode. If you have low blood sugar, or your body thinks you have low blood sugar, right? Because it can't get the sugar in. So part of the problem with diabetes is not necessarily that you have low blood sugar, but it's not getting in, right? Ultimately, your brain says, I need something. If you're not going to give me glucose, what am I going to work with? So then you're going to metabolize fats and use some of the byproducts of fat to keep your brain alive a little bit longer, but that's not good. Okay, so that's acidosis, ketosis. You're using those byproducts of fat metabolism, and so uncontrolled diabetics will sometimes smell like a chemistry lab. So you're going to smell like acetone, because one of the byproducts is acetone, and you're going to breathe out acetone because it's like a sign that things aren't right here, and we're going to try to keep you alive as long as possible, but your brain is shutting down. Never go there, please. Okay, so insulin, high blood sugar, we're going to turn on all these steps so that we're going to use the fuel that we have as sugar. So we're going to turn up everything we need to get into glycolysis. So all these layers of regulation. Again, glucagon. So we're going to see later, so the glucagon receptor is a G protein coupled receptor. So if you remember back from the signaling lecture, 2 halves of a lecture, so G protein coupled receptors, what's the downstream effect? Cyclic AMP, right? So glucagon is a G protein coupled receptor, GPCR, CR. Okay, so it's going to make more cyclic AMP, and look at that kinase. So it's a cyclic AMP dependent protein kinase, which means it's going to respond to glucagon. So now it's going to put the phosphate group on this bifunctional protein, activate the fructose bisphosphatase 2, degrade fructose 2,6-bisphosphate, downregulate glycolysis, upregulate gluconeogenesis. Okay? So have we peeled back enough layers? Okay, that's where we're going to stop on that one. Ta-da! Okay, so that is the most level of regulation that we'll see, which is important. So why is it so important to regulate phosphofructokinase 1? It's the committed step of glycolysis. So we want to be really sure we're going to do glycolysis, so we have all these layers of regulation built in to really be sure that that's what we want to do. Okay? And so finally, pyruvate kinase regulation. So where is pyruvate kinase in glycolysis? It's the last one. Okay? So pretty much once we're there, big deal. We're pretty close to the end anyway, right? So on the right-hand side of the slide, that's all tissues. Okay, so all tissues, the pyruvate kinase is regulated by kind of all the things you'd expect. So if we have sufficient energy, so as measured by downstream things from pyruvate. So we have enough acetyl-CoA, we have enough ATP, we have some fatty acids. We don't need to activate pyruvate kinase. We can get fuel from somewhere else. Okay? At this point, we're almost done with glycolysis, so big deal. It's not a major big deal. However, or not however, in addition to that, here's one of the few examples of feed-forward regulation. So fructose 1,6-bisphosphate actually feed-forward activates pyruvate kinase. So it's kind of like a heads up. Fuel's coming down the stream, get ready to go, prime the pump for enzyme activity. So fructose 1,6-bisphosphate is a feed-forward activator of pyruvate kinase. It gets everything ready to go. So that's in all cells. So we're going to say liver and muscle. So the two different areas where things happen. In the liver, so remember, the liver is a little bit different. It can do fatty acid metabolism, so doesn't always need to do glycolysis. So maybe if our fuel source is low in terms of glucose, we're just going to shut off glycolysis and do other things, do fatty acid metabolism. So it could be that we might want to regulate that differently. So just like hexokinase, glucokinase, pyruvate kinase is also regulated differently in the liver versus not in the liver. Okay, so at low glucose, we're going to phosphorylate pyruvate kinase in the liver to turn off its activity, save the blood sugar for the brain, save the glucose for the brain to use. So only in the liver does this protein kinase A phosphorylate pyruvate kinase. So what turns on protein kinase A? Glucagon. So glucagon is an indicator that your blood glucose is low. So we're going to turn off glycolysis in the liver to save the glucose for the brain and muscle. Okay? Okay. Deep breath. Yes?
So wait, you're saying that the protein kinase activated by glucagon and it inhibits...
And it phosphorylates... So it phosphorylates pyruvate kinase in the liver. The phosphorylated form of pyruvate kinase is inactive.
And that's supposed to save glycolysis because it'll just slow it down.
So you're turning off glycolysis in the liver. So the liver can use fat for a fuel source. So if you turn off glycolysis in the liver, then the blood sugar can go somewhere else.
So it'll still regulate even though it's the last step.
Yeah, because it can, it just shuts other things off. Yeah. Because remember, most of those metabolic enzymes are near equilibrium, so they're reversible. So if you have a buildup of phosphoenolpyruvate, you can go backwards in gluconeogenesis, and that will inhibit the upstream effect. So basically, if you have a buildup, even towards the end, that's going to slow everything down before that, for the most part. And so it'll just kind of dial down the activity of glycolysis in the liver in general. Yes, Carter.
Usually when people take that shot, they get sick. Is that because too much? Is that because there's too much sugar? It's not being used by the liver at all?
I have no idea. Brooke, you want to weigh in on that one?
I don't know, I think it might just be because your body's in shock. Because it's like a pill of glucagon that you put water in, and when it goes in your thigh, and I think it's just so fast that your body's in shock.
Okay.
That's why that's good.
Okay. Yeah, I don't know. Okay. The glycolysis regulation. Let's talk for 2 minutes, and then I'll let you go, or do you want to ask questions now? Let's see let me just do this last slide, and then we're not going to talk about glycogen. Okay, so we'll just talk about this last one. So gluconeogenesis regulation. So some of that we've already talked about. So the same molecules will dial down gluconeogenesis when they're dialing up glycolysis. And so the last one we're going to talk about is just pyruvate. So it's kind of sitting at that intermediate point. So once we've made pyruvate, we can keep going into the citric acid cycle, as we'll see next week. Or, we could go backwards to gluconeogenesis. So what's going to determine which direction we go? So the two enzymes that flank pyruvate, so the gluconeogenesis enzyme pyruvate carboxylase, that will take us to oxaloacetate and then to phosphoenolpyruvate. Or what we're going to see next week, pyruvate dehydrogenase complex that takes us into acetyl-CoA. So those are the two places that pyruvate can go, unless you're a yeast, right? And so it might make sense if those are regulated oppositely. Okay, so one is continuing oxidation of pyruvate and one is going backwards. So we want to regulate them in opposite ways. So acetyl-CoA, because it's the product of the pyruvate dehydrogenase complex, is going to feedback inhibit pyruvate dehydrogenase and upregulate the pyruvate carboxylase. So again, if we have plenty of acetyl-CoA, it might have come from glycolysis. It might also have come from fatty acid breakdown. So if we have sufficient acetyl-CoA, we don't need to upregulate glycolysis anymore. So we might want to shut down, the last couple of steps of glycolysis and or upregulate gluconeogenesis. Does that make sense? Okay, so essentially we have this shifting balance of fuels. So if we have plenty of glucose, sure, we'll do glycolysis all day long. If we don't have much glucose, and or we have sufficient fatty acids, we're going to do fatty acid metabolism for fuel, certainly in the liver and muscles. We can't do it in the brain, so there's this balance of fuels. Save glucose for the brain, use the fuels that we can for tissues like liver and muscle. Okay? Yes?
So I'm going to ask, this acetyl-CoA is a feed-forward inhibitor of pyruvate carboxylase?
No, I wouldn't say a feed-forward activator. Well, I guess it is. I guess it kind of primes it. You wouldn't tend to. I don't know that. So you wouldn't actually go from acetyl-CoA to pyruvate to oxaloacetate. So it's not like it's feed-forwarding.
Right, it's like not really the same pathway.
It's not the same pathway, but it kind of activates it so that pyruvate is used for gluconeogenesis instead of for entering the citric acid cycle. Yeah, so it's just like a shift. What pathway pyruvate will use. Okay, so we will end there. We're out of time anyway. And does anyone have questions for the whole group? Otherwise, you can ask me. Otherwise, I will see you tomorrow at 1. I don't know where. Let's say, as a default, room 210, which is like this upstairs lounge area, because I have a feeling that this room 8, and 207 are probably all used for class. So it may have to be 210.